of administration can be defined as the path by which a drug, fluid, poison, or other substance is brought into contact with our systems. What are factors affecting choice of route of administration? Drug-related factors, patient-related factors, and therapeutic action. The routes where a needle is used called parenteral route of administration. This table shows the most important parenteral routes used with the different drugs. Half-life can vary from biotechnology product to another. For example, half-life of TPA is a few minutes, while half-life of monoclonal antibodies reach to a few days. How can increase the circulation half-life? Enhancing circulation half-lives by using a biotechnology technique called site-directed mutagenesis or in vitro mutagenesis. In this technique, changing in one nucleotide leads in a specific gene leads to change in the encoding amino acid, and this changing in amino acid may change the sequence of a protein completely. There is a simple way to expand the mean residence time of short half-life proteins. This way includes switching from intravenous to intramuscular or subcutaneous administration. These changes are related to the prolonged residence, the nature of pharmaceutical proteins affected by tissue peptidases and leads to degraded. For example, the switching of insulin injection position from diabetics can become insulin resistant. This belongs to high tissue peptidase activity. Also, other factors that can contribute to absorption variation, like differences in exercise level of the muscle at the injection, and heat at the injection site may affect on protein nature. The state of the tissue, like occurrence of pathological conditions. High molecular weight drugs usually enter the blood through lymphatics, and low molecular weight drugs enter the blood circulation through the capillary wall at the site of injection. This figure shows the correlation between the molecular weight and percentage of lymph recovery. For biotechnology proteins, interferon alpha has high lymph recovery because it has a high molecular weight, 19 kilodalton, among the other drugs in this experiment. Oral delivery of protein drugs would be preferable because it is patient-friendly and also easy to administer. Protein oral bioavailability is very low, and this belongs to protein degradation in GI tract, and also poor permeability of GI tract wall, and this may belong to the high molecular weight of proteins. Human body has more than one type of peptidase to break down proteins, which is taken orally into amino acids, di or tripeptides. Pepsin Pepsins is a peptidase secreted into the stomach and active in pH 3 and 5. Pepsin considered endopeptidase capable of cleaving peptide bonds in the interior or in the middle part of a protein sequence or chain. Pepsin cleave peptide bonds between two hydrophobic amino acids like isoleucine, valine, leucine, alanine, proline, glycine, and the other endopeptidases are active in the GI tract at neutral pH value like trypsin, chymotrypsin, and elastase. The other part of proteases is exopeptidases cleave peptide chain from their ends like carboxypeptidase A and B. Brush border and GI lumen also break down proteins to amino acid di and tripeptides, and this considered as physical degradation. Poor permeability due to high molecular weight of proteins don't penetrate the epithelial barrier by diffusion, and the active transport of therapeutic proteins hasn't been described yet. From the previous explanation, if high bioavailability is required, the oral route isn't the best choice for administration. Accept oral immunization because only small fraction of protein has to reach its target site to elicit an immune response. Usually, target cells are lymphocytes and M cells located in Peyer's patches. Peyer's patches located in the wall of GI tract. M cells have little lysosomal degradation which break down antigen and presented to lymphocytes. Also, the density of goblet cells is reduced over Peyer's patches and reduces mucus production to facilitate access to M cell surface to improve antigen activity. Vaccine should be delivered by using microspheres, liposomes, or modified vectors such as attenuated bacteria or viruses.